Hi, welcome to our next genetics video. So far you've learned about Gregor Mendel and some of the first experiments he did to help us learn more about genetics. You've learned about the difference between a dominant trait and a recessive trait, and you've learned how to use a Punnett square to help analyze some genetics information. In this video you'll do some more analyzing with a Punnett square, and there'll be a couple questions that you need to answer before moving on to the rest of the video. There's also some more important terminology in this video that I want you to know. So let's go ahead and get started. So in our last video we stopped with this slide here, where we learned that the trait or characteristic of any organism is the phenotype. It's the thing that you can actually see, the thing that you're actually describing. We also learned that the allele combination is also known as the genotype. Those are the letters that you give to the organism in order to help analyze the genetics. And we also know that we have these alleles. We have a recessive allele, that's a lowercase letter in this system, and we have a dominant allele, which is always the capital letter in this system. Let's focus on phenotype and genotype real quickly before we move on to the rest of the video. Phenotype is a description of the physical appearance of the trait. Remember, that's the thing that you can actually see. It's the thing that you can actually describe. So in the case of our pea plants, you could say the phenotype is white flowers. That would be the correct usage in a sentence. The genotype is the combination of alleles for a single trait. This is really the genetic makeup of the organism. Again, in the case of our pea plants, in a sentence, you could say the genotype is lowercase w, lowercase w. I know that sounds kind of weird, and towards the end of this video, I'll show you a few words that you can use in a sentence like this so it doesn't sound quite so weird. So let's do an example that isn't pea plants. Let's talk about something with a backbone here. So you see these frogs, they're the same species, but in this species, the frogs can either be plain yellow or spotted yellow, and plain yellow is dominant to spotted yellow. So we can give the dominant colored frog a capital R. We could really give it any letter here, but let's go ahead and just pick R just for the sake of it. And we'll give the other frog the lowercase r. So again, plain yellow is dominant to spotty yellow, so the capital letter is the plain yellow frog and the lowercase letter is the spotty frog. But the genetic makeup, as you know, is always two letters. So the plain yellow frog, or dominant colored frog here, is capital R, capital R. It could also be capital R, lowercase r. You now know that that capital letter is the dominant trait, and it can mask the effect of that lowercase r, or that recessive trait. So these are the two possible combinations for our plain yellow frog. With the spotty frog, there's really only one combination, lowercase r, lowercase r. But for the sake of our mating here and our genetics problem, let's go ahead and choose the combination with the capital R and the lowercase r for the plain yellow frog, and of course the two lowercase r's for our spotty frog. So here's our cross down there at the bottom. So you can see we put the code letters on the square already. If you cross-reference them, there are your combinations, there are your genotypes, and of course the big R, little r frogs would be plain yellow colored, and the genetic makeup that codes for the spotty frog would be the lowercase r, lowercase r. So in this instance, a yellow frog mating with a spotty frog would give a 50-50 chance of the offspring being plain yellow or spotty yellow. And remember, that's the phenotype. So if I asked you what was the phenotype result in the offspring, you would say 1/2 or 50% plain yellow and 50% spotted yellow. If I asked you the genotype, you would say 50% big R, little r, and 50% little r, little r. Here's another one. Here's a problem for you. Here are two plain yellow frogs with the same genetic combination, the same alleles, or the same genotype, and the genotypes have already been put on the Punnett square. And what I want to know is, what are the possible phenotypes of the offspring, and what are the possible genotypes of the offspring? Okay, so here's a review of that problem that you just did. So as you can see in our Punnett square, we have our genetic combinations, and as far as the phenotype is concerned, 3 out of 4, or 75%, are plain yellow, and you can see those 3 plain yellow frogs there in the square. The phenotype also includes 1/4, or 25%, spotted yellow frogs in the offspring. This is pretty cool because two frogs that were plain yellow can produce an offspring that's spotted. This is one of those cases in which the parents that don't have the trait can produce an offspring that does have the trait. As far as the genotypes are concerned, 1 out of 4 is big R, and you can see that one right here in the upper left corner. 50%, or 2 out of the 4 possibilities, are big R. There's one right here in the upper right, and one right here in the lower left. Remember, 2 out of 4, 50%. The last possibility is 25%, or 1 out of 4, r, and that's this one in the lower right-hand corner. Take a moment to realize that the phenotype percentages don't match up with the genotype percentages. This is possible in genetic combinations, because the plain yellow frogs can be both big R and big R. So it's important to realize that the phenotype ratios or percentages will not always match the genotype ratios or percentages. Here's a human example. In humans, earlobes can either be attached, like you can see here on the left-hand side, where the skin and the cartilage just attaches right to the side of the head, or the earlobes can be free, where you actually have a lobe, where the edge of the skin and cartilage actually makes a little lobe down here at the bottom that is free hanging. And this is a human trait, and it's genetic. In humans, attached earlobes are dominant to free earlobes. So here are our genetic combinations. Let's say I chose A as our letter for our genotypes. 2 capital A's would mean attached. A capital A and a lowercase a combination would also mean attached because attached is dominant. And the only genetic makeup for the free earlobe trait is a. So here's an example that I want you to perform. We have a a, human individual, mating with a big A, human individual. I want you to use the Punnett square, if you need to, and answer this question on the right-hand side. What are the possible phenotypes from this cross? In other words, in the offspring, will you have free earlobes and attached earlobes? Or maybe just attached earlobes in the offspring? Or maybe just free earlobes in the offspring? So let's go ahead and work through this question that you just answered. Remember, free earlobe is the a, and attached earlobe is the big A. And we can put those code letters on the Punnett square, cross-reference them. And you realize that in the next generation, the offspring from this mating includes offspring that all have big A. And we know that big A is dominant, and we know that also corresponds to an attached earlobe, so we know that 100% of the offspring in the next generation have attached earlobes. This is another example in which the parent, one of which has a free earlobe, can actually produce offspring that have attached earlobes. Another example where one of the parents has a trait that the children do not. So the answer to our question is 100% attached earlobes in the offspring generation. So let's go over a little bit more terminology. Let's focus on genotype here. Let's go back to the pea plants too. So remember, our pea plants, purple is dominant. So big A would mean a dominant plant. But we use the word homozygous. Homo means the same. Zygous refers to the actual genetic makeup of the individual. So in this case, it's the same letter, big A. And we say homozygous dominant because they're capital A's, or capital letters. Another combination is big A, or a capital letter and a lowercase letter, and we call that heterozygous, because hetero means different. They're the same letter, but one is capital and one is lowercase. The only other combination we call homozygous recessive. It's homozygous like homozygous dominant, but we use the term homozygous recessive to refer to the condition where an organism would have 2 lowercase letters in their genotype. Here are the individuals from that earlobe example, a crossing with big A. In a sentence, it would make most sense to say a homozygous recessive individual reproduces with a homozygous dominant individual. And the result would be 100% of the children will be heterozygous. If we go back to our problem here, If we go back to the earlobe examples, you'll see the children in the Punnett square are all heterozygous. Okay, so what did you learn in this video? Hopefully you learned a little bit more about genotype and phenotype and how to use those words in a sentence, for example. And you learned a little bit more about how to use the Punnett squares, especially with some traits that are not related to the color of flowers in pea plants. And you also learned some new terms, homozygous dominant, homozygous recessive, and heterozygous, and also how to use those terms in a sentence.